Good evening, students. In today's class, let us we begin with types of RNA. Here, the types of RNA that will consist of so many types and their process. Here in prokaryotes and in eukaryotes, both you see here main classes of RNA molecules. Here you see mRNA, that is messenger RNA, transfer RNA, ribosomal RNA. The other RNAs are small nuclear RNA, microRNA, small interfering RNA. These are the very most important types of RNA. And also here another one RNA is heterogeneous nuclear RNA, that is hnRNA. So these are the most important types of RNA. So let us we move with mRNA, that is messenger RNA. So all members of the class function has messengers carrying the information in a gene to the protein that synthesizing machinery. Here the protein, they will carry some of the informations from one region to the another region. They carry some of the messages to the gene to the protein, and they also act as one of the important machinery. Here the terminal end is capped at the 5' end by the 7-methyl guanosine triphosphate cap. So this cap is involved in the recognition of mRNA by the translating machinery. So here the recognition of the mRNA, that is by the translating machinery, here the must and should here cap is involved. So at here region we see here the guanosine triphosphate cap, that cap is here involved in the recognition of the mRNA by the translating machinery. It stabilizes mRNA by protecting it from 5' exonuclease. Here you see exonuclease process, that is 5' exonuclease, that will stabilize this mRNA by protection mechanism. And also here the 3' end of most mRNAs have a polymer of adenylate residues. They have adenylate residues. What that residues? 22 to 50 residues, that is polymer that contains. That tail here prevents the attack by 3' exonucleases. Here the tail that prevents the attack by the 3' exonucleases. On both 5' and 3' end there are non-coding sequences that which are not translated. That is here we see the NCS, non-coding sequences. Here that is here that region, some of the intervening region between the non-coding sequences that is present between 5' and the 3' end is called coding region. So 5' and the 3' end is called as the coding region. So this region encodes for the synthesis of a protein. So here just see here. So this is the structure of a typical human protein coding mRNA, including the untranslated regions, that is UTR regions. So 5' cap region here, that is your 5' cap, that is methyl guanosine triphosphate. And also here the untranslated regions, you see here the coding sequences, and here 3' UTR, here the one of that is the poly A tail enzyme. It is very important here. 5' cap, here the 5' UTR is the start region and 3' UTR is the one is that is what coding sequences we see here up to start, that is 5' UTR start, and also stop. After that, we see here 3 UTR regions, stop regions. From here, that is 5 UTR, that is the start region, and up to end, that is 3 UTR, that is the stop region. And also you see here the poly-A tail enzyme, that is very important enzyme for the coding of mRNA, that is in the untranslated regions. And also you see eukaryotic mRNA molecules, that is 5 cap, AUG, that is the double initiated codon, that codes for methionine. Here that is leader, and also you, that is also you see here the trailer coding sequences, UAG, whereas here UAG, whereas AUG is the start codon and UAG is the stop codon. So next here you see the poly-A tail region up to 5 to 3 where the transcription site starts. Yes. So here the messenger RNA, mRNA, is synthesized from a gene segment of DNA, which ultimately contains the information on the primary sequence of amino acids in a protein to be synthesized. So that is the genetic code as translated is for mRNA, not DNA. Okay, here the genetic code that is translated for the mRNA, not DNA. So the messenger, that is the messenger RNA, carries the code into the cytoplasm where protein synthesis occurs. Okay. So here the specific amino acid has been determined by the genetic code based upon triplets of nucleotides, which provide 64 different codes using the 4 nucleotides. So here each nucleotide has a triplet called a codon, which is a triplet codon here, AUG, can be read and translated into an amino acid to be incorporated into a protein being synthesized. So all of the 64 codons or here the triplets have a known function, that is with 61 coding for amino acids and the other 3 serving as a stop or a termination signal for the protein synthesis. The other 3 are the stop codons. Out of 64, 61 are the coding codons and 3 are the non-coding codons. So there are usually several codons for each amino acid. Only methionine and tryptophan have a single codon, which is a single codon here. Only that is the methionine and tryptophan. Just see here the structure of the ribonucleic acid, that is here codon 1 up to codon 7. They have some important genetic constitutions. So that next one is transfer RNA. So transfer RNA are one of the smallest of 3 major species of RNA molecules. Yes, they have 74 to 95 nucleotide residues. Okay, tRNA that transfer the amino acid from cytoplasm region to the protein, that synthesizing machinery. And they are also called adapter molecules. Yes, you know, they are the adapter molecules. Next you see in the case of T cloverleaf model structure. Here they are also called adapter molecules since they act as adapters. Since they act as adapters for the translation of the sequence of nucleotides of the mRNA into specific amino acids. There are at least 20 species of tRNA, one corresponding to each of the 20 amino acids required for protein synthesis. Okay, so tRNA is the only RNA species that contains the nucleoside thymidine. Okay, remember this word, that is nucleoside thymidine. So you go for structure. What is the structure? That is the primary structure. The nucleotide sequence of all the tRNA molecules allows extensive intra-strand complementarity, that is complementarity that generates a secondary structure. And also you see the secondary structure, each single tRNA that shows some of the extensive internal base pairing and that acquires a clover leaf like structure. Okay, that structure is stabilized by the hydrogen bonding between the bases and is a consistent future. You see here the hydrogen bonds. You see here the adapter molecule. Here you see that is different types of. They have one of the different arms. They have that is different loops. So here the secondary structure that is called the clover leaf structure. All tRNA contains that is five main arms or loops. That is what is one acceptor arm. Okay, acceptor arm that you see here acceptor arm. Okay. Next, what you see here? What is acceptor? You know, after acceptor arm, you see anticodon arm, and you see DHU arm that is dihydrouracil. And next, what is the symbol? That is thymidine pseudo uridine cytosine arm. Also, you see here thymidine pseudo uridine cytosine arm. You see also the extra arm. Here the structure that will show some of the secondary structure of tRNA, that is the cloverleaf model structure. CCA tail in yellow, acceptor stem in purple, and you see the variable loop in the orange, and a D arm in red, and you see here the anticodon arm in blue with anticodon in black. So that is a T arm in green. So these are the important structures of the tRNA. That is here the secondary structures. See this here. Okay, you see here the ester bond formation. That is the amino acid. You see overall how here the tRNA molecule, that is some of the intramolecular base pairing are also there, and also the mRNA coding regions, that is the 5' to 3' end, and you see the anticodon regions, and that is here you see some of the amino acid binding sites, enzyme sites, which are the ribosome, that is which are the sites of the ribosomes. Anticodon, that is the mRNA recognition ends, mRNA molecules, and some of the codon that is some of the codons, they are coded at the specific positions. So the tertiary structure of tRNA. Tertiary structure of tRNA, they have what? That is the L-shaped tertiary structure is formed by further folding of the clover leaf due to hydrogen bonds between the T and D arms. So the base paired double helical stems get arranged into two double helical columns, continuous and perpendicular to one another. Here you see the tertiary structure of the tRNA. They have the L-shaped structure. So that next one is ribosomal RNA, rRNA. That is the ribonucleic acid ribosomal RNA component of the ribosome, and is essential for protein synthesis in all living organisms. The functions of the ribosomal RNA molecules are necessary for ribosomal assembly and seem to play key roles in the binding of mRNA to ribosomes and its translation. So recent studies that suggest that an rRNA component performs the peptide transferase activity, and thus is an enzyme, that is a ribozyme. So okay, that constitutes here the ribozyme, that is for the tRNA modifications. Okay, here it constitutes, that is the predominant material within the ribosome, that is which is approximately 60% rRNA and the 40% protein by weight formation. Here the ribosomes contain two major rRNAs and 50 or more proteins. So the ribosomal RNAs form two subunits, the large subunit and small subunit. So the large subunit rRNA acts as a ribosome, catalyzing peptide bond formation. Okay, remember this word here, peptide bond formation. So here this is the differences between the RNA and the DNA. Okay, RNA and the DNA, here some of the important differences are given. Just go through this PPT and you study well. Okay, we will discuss some of the important topics in the next class. Thank you.